All right, welcome to our last day. Woohoo! Evals, you have until Sunday to do those, so please take advantage of doing that. It's nice to have a large sample size of what y'all are thinking, so take advantage of that. I am not on campus tomorrow. I got some other running around things I have to do with my daughter that just got a job. But I will be here Monday. So if you want to come in, talk about anything, I'll be here most of the day on Monday. Make sure that you are not forgetting about your homework. That is due on Tuesday at test time as well. So noon here on Tuesday. So noon, not 1 o'clock. And hopefully that will go well. So we picked up, we just got influenza to finish up. We're going to talk about HIV and we're going to talk about coronavirus today. We finished a little early. I'll do a little reviewing with you. So we'll see where we're at. Last time, remember influenza virus, that's a orthomyxoviridae. That's the family of virus. These are negative sense single-stranded RNA viruses. All right, so we talked a little bit about the structure of the virion. Where we ended up last time, we were talking about, remember we talked about those two spike proteins, hemagglutinin and neuraminidase, H and N. And when those, this seems to be, we'll back up. When those spike proteins change, then that gives the virus a little maybe different transmissibility, different severity of disease sometimes, kind of like coronavirus. So we talked about the different antigenic variation that we see with those spike proteins. There's always a little bit of kind of slow mutation, point mutation, that we would consider antigenic drift. And then when we ended last time, we were talking about antigenic shift, which is a more kind of more quick change. It's more of a reassortment. And we think that this happens in China a lot more easily because in China, people are living in close proximity with their livestock. Probably other areas of the world as well, but where you might have a fully avian influenza virus and say maybe a human one, both infecting another species at the same time. So in this pig here, you could get some reassortment and end up with something that was transmissible back to humans that they hadn't seen before. So this usually accounts for more, you know, big jumps, more of our, oftentimes we think our epidemics are due to some kind of sudden change that people haven't seen before and don't have very good immunity for. So about every 10 years, the last one that we would say here was like 2008, 2009. So we're kind of due for another kind of bad flu year. So that's why you've been hearing, hey, get your flu shot, get your flu shot. We don't want to have to deal with this at the same time that we're dealing with COVID. Let's see, so transmission of influenza, same as what we have seen with some other infections. It's the large particles. Okay, this is direct contact. So typically, most often, you have to be pretty close to somebody and you get coughed on or sneezed on in some way. Less often, it can be airborne. Again, that's much less often. And also it can be through fomites. The influenza virus actually survives on fomites a little bit longer than what you would expect a membrane-bound virus. An envelope virus, we talked about how they're a little wimpy in the environment. They dry out And then once that membrane dries out, they're not going to be able to attach to the host receptor cells. So they last about a day on fomites. So that's a little bit longer. So, you know, keyboards or door handles or whatever has the potential to pass flu virus as well. Back in, we gotta at least talk about the flu pandemic of 1918 and 1919, because back then it was sort of a perfect storm of things. You had a lot of people living in very close proximity to each other due to World War I. So you had the military, you know, living in barracks, and lots of troop movement. So people moving all around the world when they wouldn't normally be doing so, especially 100 years ago when travel wasn't as easy. So it's hard for us, well, a little bit easier for us to understand now, but you know, traditionally we don't think about the flu as being something that's very deadly. But that strain back then that surfaced was different than the flu that we see today. It was much, much more severe. People very commonly would get pneumonia as a result, bacterial pneumonia. And back then we actually were pretty good at treating it, but we had to harvest antibodies from somebody that had survived it and then use those antibodies to treat somebody with pneumonia. So we didn't have antibiotics then, but we were pretty good at treating it. But when you have, you know, gymnasiums full of people, you know, healthcare system overrun, we didn't have time to harvest antibodies. Other symptoms, people would bleed from their nose. Neurological symptoms were common. They think that basically this started in the spring of 1918 and then kind of died down over the summer and just came back really full force the next fall. Woodrow Wilson had the flu, I think January or December of 1919. And when he recovered from that, he started making decisions very different than the way he previously was thinking. So that led to a harsher treaty with Germany, one of the things that they think led to World War II. So, and he died not long after that of a stroke, which they think probably the flu weakened him. So it's interesting to think about how infectious disease really dictates, you know, we can look back and see how it's, you know, the effects of it were felt in, you know, human history. Somewhere between 1/3 and 1/2 of the population of the world was infected, and about, as best we can tell, about 50 million people died. So we are not there yet with coronavirus, thankfully. Pretty high mortality. A really striking thing about that pandemic was that people that were in their prime, about 18 to 40, those were the people that were most often dying. Usually with the flu, it's the elderly or maybe the very young. We think that had something to do with those people had the most robust immune response, way too much inflammation, and that, you know, contributed to the mortality there. So the question is, was this, you know, this unique strain, was it caused by antigenic shift or antigenic drift? It was assumed for many years that it was some big shift, some reassortment in animals, something novel, but and that's still under debate. But when we look at the I guess the phylogeny, the, you know, evolutionary relationship kind of the virus, they think that it was more of a gradual drift, that some flu strain just kind of gradually was able to be transmitted to humans. So again, that will probably be debated for a long time. You might have heard it called the Spanish flu. Of course, people in Spain don't like it, called it that. They were the first ones in Spain to kind of sound the alert that this was a problem. They were not involved in World War I, so they weren't busy with other things. But we think it actually started in an army camp in Kansas. So kind of interesting. Clinical disease, you've probably all had the flu at one point or the other. Usually starts about after 2 days of being exposed to somebody with the flu. Those variants bind to the cells of the respiratory tract, cause lots of inflammation, and they bud out so quickly from our cells that they don't really, in effect, has the same effect as lysing the cells. So it kills the cells when they bud out really quickly. You probably know the symptoms, right? High fever, chills, body aches. You know, lethargy, being tired, coughing. Okay. Notice that I didn't write vomiting, remember? Sometimes, rarely, kids will vomit. I know, like one of my kids, she seems like she just throws up like anytime she's sick with anything. You know, you probably know people that are a little more sensitive that way. But remember, this is respiratory, usually not causing diarrhea or vomiting. And you have a real good case of it, it's gonna last a week or more. So it's pretty nasty. Common to have some kind of secondary infection, like an ear infection, or even to develop pneumonia, especially in the elderly. When they develop pneumonia, that's when it gets dangerous. All right, treatment and prevention. We're not really very good at either one of those things. So that's why the worry is that we're going to see another strain that's really bad, and you know, we're not really as ready for it as what we would hope that we are. The drugs, there's, I believe, only 4 drugs that are approved by the FDA right now to treat flu. 3 of them are neuraminidase inhibitors. So remember, neuraminidase is the spike protein that prevents the release. So we're going to say inhibits the release of the new virions. Okay, so I think I found this graphic that helps kind of understand what's going on here. This is from a Tamiflu ad, actually. So if you look at, let's start right here. Here's the influenza virus. It's recognizing certain receptors on our respiratory endothelial cells. It tricks our cells into endocytosing them. All right, so it's surrounded in a vesicle here. Ignore this over here. That's a drug that doesn't work anymore. All our strains of flu are resistant to that. Influenza virus is really unusual because it actually, well, it uncoats and then it enters the nucleus down here. Usually RNA viruses stay in the cytoplasm. There's no reason that they would need to go to the nucleus. There's no enzymes there or anything that they need. But for whatever reason, influenza virus does. That's where they replicate their viral RNA, synthesize their viral proteins, so that messenger RNA comes out into the cytoplasm, gets read at the ribosomes, right, to make their proteins, and those proteins get packaged into a virion over here, and then they pinch off some of the host cell membrane. They've expressed their spike proteins on that membrane, and they bud out and are released. These neuraminidase inhibitors, what they hope to do, that's what's in this box here, is to slow down that release of virion. So they are trying to keep the virion from budding off. It kind of stays attached. And if they do bud off, they're more likely to clump together. And we know that our immune system is more likely to notice big clumps of pathogen rather than just single pathogens. So it slows down viral replication. So here's the trade names of 3 of our neuraminidase inhibitors. The most recognizable one there is Tamiflu. That's been around since 1999 is when it was approved. The thing is, there's been a lot of controversy over this, but how well do they really work? Well, all 3 of them really only reduce the length of the disease by about a day. sometimes they say 1 or 2 days. So does it make sense for the government to be stockpiling millions of doses of this when it really has kind of a negligible effect? That's something that's debated. Tamiflu is available generically now, so it only costs about $50 for a dose rather than the $100 that it did before. The other caveat here is that you have to take within about 2 days of the onset of symptoms. And they are prescription only. So when you're feeling lousy, you got to haul yourself out of bed and go to the doctor just so you can get the Tamiflu. And again, kind of expensive, and maybe don't really work that well. So these are all kind of downsides of those. And with any drug, you have the possibility of a side effect. Sometimes there's vomiting or fever side effects just with taking these drugs. Sometimes people have allergic reactions to them. So again, they're not great. They are, anybody can take them though. You go to the doctor, they will prescribe it if you ask your doctor to. It's recommended for people that are high risk to have some complication from flu. So you've got some other, issue going on with your health, and maybe it's a good idea. But educate yourself and, kind of think carefully about all those things, whether it's appropriate to try to treat influenza. We do have a newer drug. This was approved in, I believe, 2018. So really pretty new. That's called Zoflooza. That's the trade name. Again, it has another name, bedaquiline, I think it is. No, it's not bedaquiline. Oh, baloxavir, that's what it is. So the mechanism of this drug is it inhibits cap snatching. Okay, so remember when we talked about our genetics, that in eukaryotes, when they, say this is the nucleus of the cell, when that DNA, oops, I don't know if I can even draw this, when DNA is transcribed to messenger RNA, that messenger RNA, before it leaves the nucleus, is going to have a 5' cap put on there and a 3' polyadenylation. Okay, so we talked about that. That happens in eukaryotes only. That's because that messenger RNA transcript has to travel. It has to leave the nucleus and it has to find the ribosome. That 5' cap is what matches up with the ribosome just right, so that translation into proteins can commence. So what Zoflooza does, what the virus actually does, it actually snatches that 5' cap and puts it onto its messenger RNA in order that it can line up right and be read at the ribosomes. Okay, very, very sneaky. So this drug inhibits the influenza virus from grabbing that 5' cap. All right, so it makes it less likely to be able to translate its own proteins. This drug is nice. It's one pill. And it's expensive. It's like $150, but studies that I have read have shown that it has a little less side effects. It cuts down, maybe 2 to 3 days on the amount of time that you're going to be ill. And you still have to take it though, right at the beginning. So you still have to take it within the first couple days. But again, it's a little bit more promising than the neuraminidase inhibitors right now. Other than that, you choose not to take any of those drugs. It's just, sleep it off. Bed, stay in bed, drink lots of fluids. Tylenol, something, not aspirin, usually is recommended, although usually we say non-aspirin, especially in kids, don't give kids aspirin. And prevention, we do have a vaccine, it's not great. we have a really robust surveillance system as to what strains of virus are circulating. There's a committee that meets in Silver Springs, Maryland every March, and they decide what are the strains that are circulating. They look at what strains circulated in the Southern Hemisphere and the Northern Hemisphere, and they decide then what strains to put in for the upcoming flu season. So it's kind of a clunky system because we have to decide that so far ahead of, October, November, December, when our flu season is. So, hopefully we can advance that. The reason they have to do it so far ahead of time is because most of our flu vaccines are grown up in eggs, and that takes a while. They have to then deactivate it. Lots of controls in place to make sure that that happens. So if you hear somebody say, I got the flu from the flu shots, it's not possible. It's an inactivated virus. There's some purification that goes on. There are many different kinds of flu vaccine. If you're allergic to eggs, you can get a recombinant version that's not grown up in eggs. And we actually think that version may be a little bit better for some populations. There is an attenuated version that tends to work a little bit better in kids of a certain age. That's a strain that only will replicate when the temperature is a little bit lower. So you breathe it into your nose and it replicates in your nose, but then can't replicate in the chest. So only very mild. All right, so it is, recommended to get the vaccine. On good years, it's only about maybe 60% effective. It all depends how well they do with matching up the strains that are circulating at that time. On a bad year, it may be more like 35 or 40% effective. But again, if you can reduce your chances of being out for a week with the flu, that's something that I'll jump at. So sometimes from year to year, some of the strains stay the same, but there's usually a little bit of change there. All right, questions about influenza? All right, so HIV. It's actually in the genus Lentivirus, which is maybe not as recognizable, in the family Retroviridae. So any virus that can make reverse transcriptase, we'll talk about what that is in a minute, is in that Retroviridae family. Another example, hepatitis B, actually, I haven't really talked about that one very much. But HIV stands for human immunodeficiency virus. It's another enveloped virion, and it has a segmented plus sense single-stranded RNA. So it actually comes with 2 segments of that messenger RNA. They're copies of each other. Okay, so 2 copies of that strand of messenger RNA. And also included in that virion, and you can kind of see it here, here, and here. It also has 3 key enzymes that are carried in that virion. The first is reverse transcriptase, which I just mentioned. Okay, when scientists discovered this, it was really one of those mind-blowing things, because what this enzyme does, let me write it up here. So again, messenger RNA is single. Remember, positive sense is what can be read like as messenger RNA. So what reverse transcriptase does is to make single-stranded DNA from that messenger RNA. So right, that's something that we never do. That's against the flow of information. And then from that, it will copy it and make double-stranded DNA. And then from that double-stranded DNA, it makes messenger RNA. All right, so it seems like a very circuitous path. It could just make a negative sense messenger RNA, and then from that make more plus sense messenger RNA, but it goes through a DNA intermediate. So very strange. Why does it do that? Well, double-stranded DNA is a little bit more stable. And also it does integrate into our chromosomes. So it couldn't do that unless it made into DNA. So Again, I'll write this here. It's an enzyme that makes DNA from RNA in a nutshell there. Okay, two other enzymes, and these become important as we think about how a drug's designed to treat this disease. Integrase, which helps it to integrate into host DNA, and protease. This is necessary, we'll say, for maturation of the virion. So basically, this protein has to come in and make some cuts in some other proteins or enzymes in order to make them functional. Okay, so we'll get back to that in a minute. Other parts of the virion that I want to draw your attention to, there's a few key glycoproteins. So GP120, glycoprotein 120, that's important for attachment to the host cell. And GP41, we think is important to fuse the viral envelope with the endosome that it gets brought into the cell with. Okay, so this is a nice graphic from your book that kind of illustrates what all those things are doing. All right, so here's the HIV virion. It's using these little kind of tabs here. That's the GP120. The GP120 actually recognizes a glycoprotein on our cells, CD4. So that should ring a bell. What kind of cells have CD4 glycoproteins? T helper cells. Okay, remember, it's the T helper cells that have CD4 and the cytotoxic T cells that have CD8. It turns out that there are, and we didn't talk about this before, it turns out that there are other cells that have those CD4 receptors. B cells and macrophages have them as well. So HIV can infect B cells and macrophages as well. You never hear about that though, because infection doesn't seem to harm those cells for some reason. It's the CD4 helper cells that it really decimates. And this is a problem because remember, the helper cells are necessary to most of the time activate B cells to make antibodies and to activate cytotoxic T cells to kill virally infected cells. So without those helper cells, your adaptive immune response is pretty low. So we'll talk more about that in a minute. So it's attaching to those CD4 receptors. It, again, tricks the cell into endocytosing it. So it's bound in this membrane for about 30 minutes or so. And then we think that GP41 will kind of allow the viral membrane to fuse with that endosome membrane and kind of uncoat it. The capsid opens up, and then we've got those three proteins as well as the double-stranded DNA. Sorry, the messenger RNA. And then the reverse transcriptase makes that that single-stranded RNA into DNA. And the DNA enters the nucleus, where with the help of that protein integrase, it integrates into our host chromosome. All right, so now it's correctly called a provirus. Remember, when bacteriophage integrate, we call it a prophage. When when viruses do that in animal cells, we call that stage a provirus. Remember, herpes virus also integrates, but in a little different way. It forms episomes. Remember, little plasmid-like structures in the nucleus. Doesn't actually integrate, but essentially it's the same thing. They're always there. All right, so it integrates into the host chromosome. It can transcribe back into messenger RNA and translate that into the virion, assemble it, release. At some point, that protease works. And I looked this up a while ago, and I think your book makes it sound like it happens once it comes out of the host cell. Other sources I read makes it sound like that happens when it's still inside the host cell. Whatever point, that protease is necessary to activate certain enzymes. So make cuts at certain places so that they are functional. All right, so we call that kind of the maturation. The virion's not mature unless that has happened. All right, so when we think about those three enzymes, especially that are necessary for the replication of HIV, that makes us think, well, what drugs can we tailor to act on things that the virus has that we don't? So that selective toxicity, all the drugs in some way, I believe, will target one of those three enzymes. So sometimes they target reverse transcriptase. And our early drugs, AZT is kind of the famous one. It's a nucleoside analog. So we talked about that several times. What was the nucleoside analog that was used to treat human herpes virus? Acyclovir. Okay, so it's kind of similar to that. This one actually mimics thymine. So this is thymine base. Deoxyribose looks very similar, except it has an azide group here. So when that's incorporated, when reverse transcriptase, really incorporates lots of different things. It's not picky about what incorporates into its new DNA strand. It will grab that, incorporate into the chain, and then it terminates replication because, again, there's no 3' hydroxyl group here. So that causes chain termination. And there's lots of other nucleoside analogs as well. I didn't indent this correctly. There's also drugs that target integrase and also drugs that target protease. And I didn't really get into the mechanisms there. Usually patients will take a combination of at least maybe 2 or 3 or more different drugs just to kind of make sure you are trying to keep that virus from replicating by lots of different methods. With any drug, there's possible side effects, and these are no exception. Usually when a patient first starts taking them, the side effects are the worst. And then over time, they generally get a little bit better, but it's very common to have some nausea, fever, insomnia. The list of possible side effects goes on and on and on. There are more serious ones that can carry some more long-term side effects, things like cancer and just about anything you can think of. Question? So why is it critical to have a 3' hydroxyl group? That's where the new nucleotide adds on, right? So it's, there's another phosphate up here, right? And that's added on to the 3' hydroxyl group of the next base. Okay, so good question. Okay, so lots of side effects, but people are living a lot longer with HIV now, and we'll talk about that in just a second. Transmission, probably aware of this. I can remember 80s when this came out. There was a lot of just wild hysteria about transmission. There was rumors that you could get it from sitting on a toilet seat in a public restroom or swimming in a public pool. I was thinking today, where did we get our information back then? That was before the internet. It's kind of hard to imagine now. But we know that HIV is transferred by very direct methods, not airborne. You have to have, not droplet spread, thank goodness. Either through some bodily fluid, like a blood transfusion, through sexual intercourse, IV drug users by sharing needles. Mother to child, about 1/3 of babies born to HIV positive mothers are HIV positive. So it's not a for sure thing. It's likely, but not a for sure thing. It can also be transmitted through mother's breast milk as well. This is not something that you're going to get by exposure to some fomite. It's really wimpy in the environment, degrades really quickly in the environment. Okay, you can drink HIV and it's not going to get you sick. Okay, so it has to be kind of the correct portal of entry there. Numbers, this was the latest numbers that I could find. Still, 37 million people worldwide with HIV, about 1 million of the US as of 2019. Sub-Saharan Africa has really been decimated by this. About 5% of sub-Saharan Africa people are HIV positive, and there are pockets where it's more like 30%. So really still a huge problem, but as of 2011, life expectancy was 70 years. And today, I just looked this up a few days ago, it said normal lifespan. So that's pretty astonishing. In 1996, the lifespan was 39 years. So as long as patients get on these drugs, the sooner the better, after they know that they're positive, and they stay on the drugs, they stop taking the drugs, and then the virus can start to replicate again. As long as they stay on their drugs, it's likely that something else will kill them, like a heart attack or cancer, and not complications from HIV. So it's really astonishing. What's going on in the body? This is a really kind of a cool, complicated figure in your book here. What happens when somebody's infected with HIV? So let's start here at the number 1. This green line is the CD4 helper T cell count. So when a person is first exposed or infected, the variants target this helper T cells, and a lot of them die really quickly. All right, so the green line is this these units on the left here. Okay, at the same time, so the red line here is the virions in the blood. So the CD4 cells are dying, and we got lots and lots of viral replication. But our immune system, at least to begin with, is able to mount a little bit of an antibody response. And this purple line here is the amount of antibodies. And so usually after that initial infection, things kind of, the HIV goes latent, usually for months. I don't know if there's a average time span there. If there is, I don't know what it is, but usually it kind of goes quiet for a while. Okay, and this is with no treatment. But eventually, the virions will start replicating again. We get kind of a slow uptick here in HIV virus titers. And at the same time, the CD4 count is getting lower. The body's less likely to be making antibodies there. And when the CD4 count is below 200 per cubic millimeter in the blood, then a person is considered to have AIDS, so acquired immunodeficiency syndrome. And so then without the antibodies, the virus is basically, replicating just without any stops. And eventually a patient will die not from that virus multiplication, but from the opportunistic pathogens. So things that wouldn't normally make a healthy person sick are, the person just has no defense to. So here's a list from the CDC of the kinds of infections that AIDS patients will get. If a patient has one or more of these infections, then they're considered, I think, or maybe it's, if you have HIV and one or more of these opportunistic infections, then you're considered to have full-blown AIDS, no matter what your CD4 count is. You'll recognize some of the things on here. There's a few of these things that a healthy person can get, like tuberculosis, but somebody with AIDS is a lot more likely to get tuberculosis. And there's a lot of like fungal things or, things that, again, a healthy person wouldn't normally get. So that's a big red flag that, a person is HIV positive. Okay, but again, the drugs really are really amazing, and people are living long life. That drug regimen costs about $20,000 a year. So, and that's an old number. It's probably more than that now. So it is expensive, but okay. Any questions about HIV? All right, so we will move on to Coronaviridae. So we're going to talk a little bit historically about some of the other things that coronavirus can cause before we get to COVID-19. Really, a few years ago, if we were going to talk about this in class, I'd say the most interesting thing about it is it causes the common cold. Okay, rhinovirus causes a lot of colds, probably more than coronavirus, but and there's several other things that can cause colds, but that's kind of what it was known for more so a few years ago. It's a positive sense single-stranded RNA. Okay, so it can just act as messenger RNA. It's envelopes. And some examples. So SARS-CoV. So these are the names of the viruses and the infection that they cause. SARS-CoV is severe acute respiratory syndrome. And we'll talk more about that in a minute. There haven't been any cases of that for almost 20 years, but we'll talk about that. MERS-CoV, that stands for Middle Eastern Respiratory Syndrome. There are still new cases happening of that, but it's not very common. And of course, SARS-CoV-2 causes the infection known as COVID-19 because it was first isolated in 2019. And when we sequence the enzyme that it needs to replicate its RNA, RNA-dependent RNA polymerase, we saw that the sequence was most similar to SARS-CoV. So that's why we named it SARS-CoV-2 instead of MERS-CoV-2. Okay, so again, looking at that genetic analysis is really important and can give us clues as to where reservoirs are and kind of where it came from. So that phylogenetic analysis. All right, so a little closer look at the virion. I couldn't really decide which graphic was better, so decided to put them both up. It's an unsegmented genome. So we've got one big long string of RNA, and it's actually got the largest RNA genome of any RNA virus. Okay, it encodes for 29 proteins. Okay, so apparently that's big for an RNA virus. It has a helical capsid around that genome. The virion itself usually looks round just because it's enveloped. So a couple of the structural proteins that we'll talk about. Of course, we have to talk about the spike proteins. That's responsible for fusion and attachment to the host cells. That's the part of virion that's really the most antigenic, meaning what our antibodies are most likely to recognize. So it gets a lot of attention. Most of the vaccines are targeted towards those spike proteins. They're also the part of virion that changes the most quickly. So that can be a problem. Those spike proteins actually bind to, you might have heard this, ACE2 receptors in our body. So ACE2 stands for angiotensin converting enzymes. Okay, those receptors that we have in our nose and in our lung are necessary to help us regulate blood pressure. Okay, so those spike proteins recognize those ACE2 receptors. One other protein that I'll mention here, there are a few other ones. They just call it an envelope protein. That's, we'll just say assembly and release. So there are a couple of different proteases that we're going to talk about soon as well. They are not carried in the virion like HIV does. They are transcribed early on from the messenger RNA. Transcribed and translated from that messenger RNA. So we'll talk more about those protease enzymes shortly. Reservoirs, where did these coronaviruses come from? We heard a lot about this in early days of the epidemic, about these wet markets in China and Wuhan, because two of the people that were first kind of discovered to have COVID-19 were workers in these wet markets. So wet markets are places where they sell all kinds of food, fruits and vegetables, as well as butchered meat, as well as live animals, because in that part of China, it's really culturally, how do you say it? It makes you look good if you eat exotic animals. So all sorts of animals from all over the world are brought into these wet markets. They're stored in little cages. They're often on top of each other, so their feces rain down on each other. So kind of tight conditions. And it was assumed that those workers, picked up some infection from some of these animals from somewhere. Coronaviruses are zoonotic. They exist in all different kinds of animals, but typically they don't pass from one animal to the next. So, horse coronaviruses pass between horses, and cow coronaviruses between cows. So only rarely would they cross species barrier. So if we look at that SARS outbreak in China, okay, so 2002 and 2003 in this southern province of China, which is a different place than Wuhan. I looked that up on the map. They all told had about 8,000 cases, 700 deaths. So mortality rate about 9%. So that's definitely higher than COVID-19. People over 60 were hit pretty hard. The outbreak did spread to a couple other countries, but once the Chinese government kind of admitted that there was a problem going on, they put mitigation efforts in place that was able to stop those outbreaks. Because unlike COVID-19, people weren't contagious until they showed symptoms. So, you're sick, you stay home, and then it doesn't spread. Let's see, what was the other thing? I'm blanking out. So it was just a little bit less transmissible as well. So not terribly transmissible. If you didn't have symptoms, then you weren't transmissible. So that helped stop that. The reservoir here, they traced this back to, as a primary reservoir, to the lesser horseshoe bat. Oops, not hat. I don't think I spelled horseshoe right. That doesn't look right. Let's try that again. Okay, this is just a little tiny bat, would fit in a palm of your hand. And they think that the bat coronavirus just kind of made the leap, kind of spilled over into a human that perhaps came in contact with it, maybe at a wet market, maybe some other time. An amplifier host or a secondary host, they have identified as the civet cat. Right, so there's a picture of civet cats. They are considered a delicacy in China. Again, like I said, there's a saying in that part of China, they will eat anything with wings that's not an airplane, and anything with legs that's not a table. So civet cats, they're actually farmed in big farms in order for people to eat. And they think that the virus just kind of amplifies. So maybe civet cats out in nature are more likely to come in contact with bats. When the civet cats get sick, they just, transmit a large amount of virus. Symptoms of SARS is pretty similar to COVID-19. Fever, dry cough, headache, muscle aches. Death is usually due to respiratory failure. Most of the cases were healthcare workers. So somebody was very ill, they went to the hospital and they didn't really know what they were dealing with. And that sick patient would probably infect many other healthcare workers. Okay, again, and this faded after, they could really clamp down. We were hoping that the same thing would happen with COVID-19. So when it first came into the country in early 2020, everything stopped, right? Everything locked down, and we hoped that that would be the end of it. And of course, that wasn't the case because COVID-19 is a little bit different clinically. MERS, Middle Eastern Respiratory Syndrome, that emerged in 2012. Another respiratory infection. Most of the cases have been in Saudi Arabia. 2,400 cases as of 2019. I looked up yesterday, and actually there's been about 150 more cases since then, and about 30 more deaths. So this is still an ongoing thing. Case fatality rate of 35%. So this is a more serious infection, more often fatal. The reservoir for this is also bats, which is kind of surprising because you don't think about there being bats in Saudi Arabia. I don't know, there probably are. More importantly, we know that the viral load is amplified by dromedary camels. So it's the Bactrian camels that have the two humps. The dromedary ones, which they use all over the Middle East, have one hump. And most of the cases are people that have come into contact with camels. People can spread it to other people, but it's less likely for that to happen. So more often, if somebody was in the same family or maybe a healthcare setting, that might happen. So what they have done to try to mitigate the spread of this is kind of regulates the movement of camels around the country. Camel handlers have to wear PPE to protect themselves. And there's also been laws about drinking unpasteurized camel milk, and also laws against drinking camel urine. Apparently that's a cultural thing there. So if you don't believe me, there's an article there, you can read about that yourself. So they're trying to make it less common. Symptoms, it's respiratory, but then other parts of the body can be affected in serious cases, like the kidneys can shut down, the pericardium can be affected. No vaccine, either to SARS or MERS. It seems like maybe they could be working on one that would be similar to the COVID-19, but because there are so few cases, that's not something that probably has had a lot of money dumped into it yet. Okay, so that moves us to COVID-19. And everybody's tired of hearing about this. I'm sure you already know a lot of this already. I thought it was kind of interesting to look at the deaths, like every time that I have taught this the last few semesters. So December 2020, over 280,000 deaths. And then 5 months later, we had doubled that in the US, the number of deaths. And now, whatever, 6 or 7 months, 8 months after April, we have not doubled it. So of course, the vaccination plays a big role there, in slowing down the deaths, but now we have a strain that's a little more transmissible. That's the common strain right now. So they're saying that boosters are really important. So put that on your list of things to do maybe over the holidays is try to get that booster shot. You all know the signs and symptoms. Probably the most kind of striking one that's different from a lot of other infections is that loss of taste and smell. transmission, again, mostly respiratory droplets, can be airborne, can be further than 6 feet, more rarely can be spread by fomites, but we don't think that's very common. So, you know, early pandemic, they were saying, leave your groceries in the garage for a few days before you bring them in the house. No, not necessary. Like, it's not commonly going to be spread that way. So that's not something that we would need to do. So this is what you have when you have a mild case of COVID-19. When you think about what kills people, there's all sorts of things. And I found a good article about that I didn't find in time to post, but if you're interested in that, I can send it to you. Pulmonary embolisms, so inappropriate blood clotting. It affects the heart and can cause heart attacks, again, the clotting. Viral pneumonia, lots and lots of inappropriate immune response, so lots of edema in the lungs. It can affect the kidneys. It can affect your gut. It can cause, you know, diarrhea and vomiting. There can be neurological symptoms. I was talking to somebody, and we were talking about a treatment for somebody she knew. She said, "Oh, we're not going to take that drug because I heard that makes your organs shut down." COVID can make your organs shut down, so not necessarily the drug. I'm not sure how would you distinguish, but it can cause shock, your blood pressure to go way down, and yes, your organs to shut down. So I think probably we all get tired of people that kind of take their information anecdotally. My mom had it, she was okay, so I'll be fine, right? So as scientists, we need to pay more attention to studies with a larger sample size and not just the people we know, and kind of understand like what the experts are telling us and looking at statistics and kind of going more by that. So reservoir, I'm not going to spend a lot of time on this, but I thought this was interesting. We think maybe it's bats. We haven't really made much progress on figuring that out because the sequences, the evidence that should be there in bats, and there's a lot of different bat species, we have not found. So that begs the question, this lab in Wuhan, this virology lab in Wuhan was doing what's called gain of function studies. So basically what they were trying to do with coronavirus was make it more infective. And actually, we shouldn't necessarily blame them because their funding was coming from somewhere in the US. So it kind of, there's a lot of evidence to point to this virus could have come out of a lab. Of course, the Chinese don't want that evidence to come out, and we'll probably never know. But it makes you think maybe we shouldn't be doing those kind of studies anymore. Maybe there should be a moratorium on gain of function studies. Again, we probably won't know. Early on, they said maybe the pangolin are possibly intermediate hosts. It may be just that they are able to get the virus as well, that they're just hosts. I just included this picture because it's such a cool looking animal. It's a mammal, but it's got scales on it like a reptile. They're not extinct, endangered. It's an endangered animal. Probably, you know, there in the wet markets. All right, let's talk a little bit about variants before we talk about some new treatments coming out. So that enzyme that the virus uses, RNA-dependent RNA polymerase, it makes a lot of errors. So we get really high mutation rates with those spike proteins. So it makes sense that we're going to be having, you know, kind of different versions of the virus as we go here. The WHO, the World Health Organization, decided early on that instead of naming the variants by where they were kind of discovered, that it would be more, you know, we don't want to offend anyone, so we're going to call these variants by Greek names. All right, so the alpha variants, that was first discovered or sequenced in the United Kingdom, I think at a lab in Oxford. So we were calling it the UK version variant for a while, if you remember. And that was in November of 2020. Okay, so that was kind of the first variant of concern. They estimated that the R0 was about 2.5, so a little bit more transmissible, perhaps just a touch. Remember, we talked about R0 last time. That's if everybody was susceptible to the disease, that's the number of people a sick person would likely to infect. So maybe you know, hear different R0 calculations from different sources, but that was perhaps a little bit more transmissible, the first variant of concern. And then the Delta variant is the one that's the predominant strain right now. That was sequenced or discovered in India just a month later, December 2020. Okay, so that's the variant in circulation here now. Okay, and this has been even more transmissible than Alpha. I think last lecture I said the R0 was about 7. Other sources put that at more like 3 or 4. So I'm not really sure what to call that, but we think that with the mutations, I think it has about 9 mutations in the spike protein from the original one that have made those spikes more likely to stick to those ACE2 receptors on the host cells. They adhere more tightly, and hosts tend to just have a higher viral load than the wild type. So again, that's the dominant species now. So now we're starting to hear about this other variant, Omicron. So a little lesson in our Greek alphabet. Those of you in a fraternity or sorority probably know this better than me, but it kind of comes in handy when you're doing crossword puzzles and stuff. Okay, so we've had Alpha. We've actually had all these other variants that we've just kind of either forgotten about or they didn't make much of a splash. You might remember the Mu variant they were talking about for a little while. So the next variant should have been named New, the new variant, which the WHO thought might be confusing to call it the new variant. So they didn't use that one. And then this XI, I don't know, we would pronounce it like the beginning of xylophone, Xi. But in Chinese, they pronounce that Xi, and that's a very common surname in China. The Prime Minister of China, Xi Jinping. So because it's a common name in China, they decided, oh, they don't want to offend anyone in China, so they didn't use that one. So that leaves us with Omicron. A lot of controversy on how to pronounce that. So there are many correct ways, apparently, to pronounce that. I'll call it Omicron. So that's kind of the big controversy there. So this was first sequenced in South Africa just last month, so November 2021. In South Africa, they're sequencing a large number of their positive cases. So it may not have originated there, but that's just where we first figured out or found it. We're still trying to figure out how to treat this variant, but it does have 32 mutations in the spike protein from the original cell, so from the original virion. So it has a lot of changes. Early data says that this is even more transmissible. But perhaps a little bit milder disease. And again, early days for that, but every day they seem to be a little bit more sure that this is going to be a little bit milder disease. So that seems good. At the same time, if it's really transmissible and you have more people getting it, you're still going to have, even if it's a small percentage of people with serious disease, that could still overrun our health system. So they are saying that with the booster, your antibody count goes up higher, and should have a good chance of being protected. Even with Delta, about 1 out of 10 people that are exposed will have a breakthrough infection, even if you've got 2 vaccine shots. So a lot of you have experienced that. So again, good idea to get your booster shot if you can. Let's see. They are having really just an explosion of cases in South Africa. So that was kind of really caught the attention. So don't know anything about R naught yet or anything like that. So let's talk about treatment. So I did a lot of updating here from last semester, thinking about treatment. You know, in the past, a lot of supplemental oxygen for patients, ventilator when that gets serious. We're having a lot of success using monoclonal antibodies. So monoclonal antibodies are made in a lab and they're fashioned to fit those spike proteins. These have to be given by IV. They are not just doing that in hospital now, they are going to people's homes to do it. And it is an effective treatment. The problem is, as we get these variants with different spike proteins, we're going to have to be nimble with our monoclonal antibodies and making antibodies that fit those. Nucleoside analogs actually have the potential to be a good broad spectrum drug, meaning that the hope is that we might have some kind of nucleoside analog that would be good for lots of different viral infections. We don't quite have that figured out yet. The one that's been, nucleoside analogs actually can work in several different ways. So the way acyclovir works is the same way as remdesivir works. And that's, I just put the company that manufactures that, Gilead. Remdesivir has been around for a while. We used that to try to treat Ebola patients. It didn't really work very well for Ebola patients. We're finding that it doesn't work very well for COVID patients either. But it was something that we knew was safe, so it was worth trying. These, like I said, they're IV injected, and they are going to, the RNA-dependent RNA polymerase is going to incorporate them, and then that causes chain termination. So just like the acyclovir, just like the AZT HIV drug. Okay. Again, the benefits of that are not statistically significant. The new one that has just kind of, I think they're on the brink of, the FDA is on the brink of recommending this, is molnupiravir. So this is a nucleoside analog, but it works in a little bit different way. When it's incorporated by that RNA-dependent RNA polymerase into new messenger RNA strands, it doesn't terminate the chain, but it actually, when that new chain is used as a template to make more messenger RNA, it doesn't match up the bases correctly. So basically, you get many mutations building up and what they call like error catastrophe. Okay, so let's see if I can write that a little bit more succinctly. We'll just say mutations accumulate. Okay, so not a chain terminator, but lots of mutations. So when the FDA advisory committee looked at the data for this, 13 voted to approve it and 10 voted not to approve it. Because maybe they're saying it cuts down serious disease or hospitalizations by 30%. So a good portion of the FDA advisory committee said the benefits of this do not outweigh the possible side effects of it. And when what they were talking about side effects, you know, basically this is a mutagen. So do we really want to speed up the evolution of COVID-19, you know, COVID SARS-CoV-2. Maybe not, maybe we don't want to do that. And there's the potential that it could be incorporated into our cells that are rapidly dividing. So that would mostly be fetal tissue. So we have to make sure that pregnant women don't take this, again, because it has the potential to be a mutagen in our cells. The advisory committee also found fault with some of Merck's experimental design and their controls and things like that. So I don't know if that's ever going to take off, you know, maybe use it in kind of extreme circumstances, but both of these need to be given in early days of infections. So usually maybe the first 4 days or so. And a lot of times people don't seek any treatment, so they're way beyond that. Once somebody is in a hospital with serious disease, then usually it's not the viral replication that's causing the problem, it's all the immunological, you know, inflammation, the consequences. So that's why you need to give it early on. And this is actually a pill, so it's a little bit easier to administer. But again, there's some downsides to this. The really drug that has a lot of potential, and I found a couple of good articles that talk about this. This is a review in Nature that compares molnupiravir with Paxlovid, which is the newest one. This is a protease inhibitor. And this is actually from Pfizer. This link here, it's a little 2-minute animation of how Paxlovid works. So I usually wouldn't go to a drug manufacturer to really get, you know, good information on it. I'd rather get it from the CDC or something, but that's a really nice animation. Paxlovid is a protease inhibitor. So we're building on 20 years of protease inhibitors that we've used for HIV. And this is a very, you know, specific to the COVID protease. The protease enzymes have a very slow mutation rate. So this has the potential to, you know, work for a long time. I think the numbers here that it's like 89%, okay, cuts hospital infections by 89%. So, you know, I think they actually had to halt their study. You know, they were giving half the people Paxlovid and half the people some placebo. And at some point, it's not ethical to not give everybody the drug. So cuts hospital or death rate by 89%, and it's oral. So it's a pill. That's a really good thing. So basically what the protease does, and this animation does a really good job of explaining this, that enzyme, RNA-dependent RNA polymerase, it actually, when it gets transcribed, it's in this big, long protein, but it's not functional until it gets cleaved like 13 or 14 times. So there's 2 different proteases that have to cleave that enzyme so it's functional to work and start, you know, able to replicate the virion. So if those proteases aren't cutting that enzyme, it's not functional. All right, so we'll say, if I could write that out, it inhibits formation of that enzyme, RNA-dependent RNA polymerase, necessary to make more messenger RNA. Well, RNA, yeah. Okay, so there are lots of other similar drugs kind of in the early phases of testing. So I think it's not going to be long till we have more choices to treat. So along with good testing, you got to find out early that you have this so you can get on the drug early. This one also, you need to take early days of infection, combination of vaccine, good testing, and some drugs. I think there's a lot of hope that we won't be wearing masks forever. All right, so I'll leave you with that. Again, I guess we didn't have time to review very much, but if you have questions or anything like that, again, I'll be around most of Monday. You can send me emails. Have a good weekend. Oh, did I forget? Oh,